I mean, you all know Rachel, of course. Rachel and I've been working together for the past, I think, 4 years, right? And she came from UCSD, from a group that I've long admired, Mark Myers, and many of you will know Mark. And I think Rachel came at a really interesting time, and you'll see this today. So the beginning of Gen AI being useful for science and engineering, and I think you've done a really great job in leading this transition. Written many papers, really influential MIT Gen AI Consortium white paper, which I think we'll maybe hear a little bit about. And yeah, so my great pleasure. I won't say much more, but the work speaks for itself, and I'm really excited to learn about sort of what your overall conclusion is and how you're putting it together today. So thank you very much, and thanks all for coming.
Okay. All right. Thank you all so much for being here. I mean, as you probably know, my name is Rachel Liu, PhD candidate in the Department of Material Science and Engineering at MIT. This is my thesis, Biological and Bio-Inspired Materials Design through Generative AI Approaches. So just as a quick overview of what we're going to talk about for the next 45 to 50-ish minutes. I'm going to do a brief little introduction into what biological materials are, what's Gen AI, why biomaterials in Gen AI, and then I'm going to hop into 3 core research projects for the next 3 chapters, and then I'll wrap up with just a really brief conclusion. So without further ado, actually a few more notes I want to say is that I'm not going to be able to show every single experiment that I did here, but I'll show a brief highlight reel. I'll try my best to make it inclusive for all the folks here as well. But if you have any questions, I'm also happy to take them during the public questions or also afterwards. So without further ado, let's hop into the introduction where we can talk about biological materials. So biological materials are structural natural materials that are found often in nature. They're found in plants and animals, such as things like coconut shells, armadillo shells, bamboo. And the reason why we study these materials is because they are evolutionarily optimized. They have had billions of years to be optimized for survival. In the case of spider silk, this is a material that's heavily optimized for habitat building, capturing prey. And because of this optimization, they're particularly good at kind of having these really interesting mechanical properties. Kind of mechanical properties is my focus because I'm a mechanical engineer at heart. So there's many other properties, but particularly we're going to focus on mechanics. And on top of that, spider silk is often described as being, stronger than steel per density. And the reason why this is the case is because a lot of these biological materials have these very intricate hierarchical structures, which mean that these structures across length scales have different design elements that all contribute to that microscopic behavior. Okay, I'm going to better explain that in the next slide. What are those design elements? This is kind of pretty much research has shown like these are kind of the 8 most common design elements that you'll see throughout nature. And they'll exist across all different length scales and also in many different combinations throughout nature. For instance, the pangolin shells has these overlapping layers. The horsehoof wall has a tubular gradient. And the mantis shrimp exoskeleton has this bulligon-like structure, this helical layer fibers. And you can see they exist not only, in combination, but also across length scales. All right, but let me back up a second. So biological materials, again, why are we studying them? I already said that they're good at kind of having these interesting multifunctional structure properties. They're evolutionary optimized, but something that's really particular about biological materials is that they're naturally embedded within native ecosystems, meaning that they naturally decompose at end of life, and those materials are recycled. And when you compare that to the human material ecosystem, we really shy in comparison, where oftentimes we design materials with properties in mind and give really less consideration to end of life procedures for materials. So without that, 2 points I want to make about just the field of biological materials. It's a pretty ample amount of data that's been collected in this field. Marcus already talked about Mark Myers, who's been, working in this field for decades, probably longer than I've been alive, on studying all these different types of biological systems, things like bird feathers, the sea sponge, crab exoskeleton. A lot has been done in this field. And particularly that was actually kind of confusing when I first started my PhD because I was like, what do I study? Like a lot has been studied already. And then the other aspect I want to point at is also this potential design space for bio-inspired engineering. So, not only we study these biological materials, we oftentimes can also see kind of these representative examples of how we could use biological systems to inspire engineering applications. In the case of architecture, we have the Gherkin Tower in London, UK. You have the Eastgate Center in Zimbabwe, which takes inspiration from termite mounds to help with insulation, and a few other examples. But that's to say, though, that, this isn't a lot. This is just four. There's more too, but this is just four. And I would argue that there's this kind of growing gap between the amount of knowledge that we have about biological materials and how we could actually apply them in scalable bio-inspired design. And so, with that being said, we're coming back to this figure. We're going to think about, okay, well, we should improve our own human material ecosystem. And one way that I suspect that we could do this is by learning from nature. And I think we can do this in two ways. First way is either engineering directly with biological materials themselves, using abundant feedstock material that's already naturally degradable. And then the other end of this is also designing higher performance synthetic materials that are inspired by biological design motifs. And the main hypothesis of my thesis here is that we could accelerate this by using generative AI. Okay, sound good? All right, so generative AI, I know, very hot topic. Artificial intelligence has been around for a very long, long time. Generative AI, obviously very new. It's really, really popped up in 2023 as this kind of subset of artificial intelligence. Within the context of scientific like research, classical machine learning is really more focused on kind of this recognition, this understanding the relationships between inputs and outputs. What really sets generative AI apart from other artificial intelligence methods is this creation method, where instead of kind of just understanding relationships between inputs and outputs, we're instead having these generative models that are able to produce new and novel content. So, for instance, we're able to train a generative model on a mass amount of data, and that generative model could produce new data that we have not seen before. So, particularly, I'm going to focus on the attention operator. The attention operator in Transformer was introduced in 2017, and this is what powers a lot of generative models nowadays. And what the Transformer architecture enabled was that it allowed models to understand not only short-range interactions, but short and long-range interactions. And what that means for natural language processing is that words and sentences, we're able to understand this better, or models are able to understand this better because words not only can attend to the words to the left and right of it, but they're also able to attend to words later on the sentence as well as itself. And because of this, this had led to the rise of language models. Language models have been around for a long time. Those models are really good at telling, is this email, is this person angry, typing this email and stuff like that for sentiment analysis. But what really sets large language models apart is that they're primed for generating new text and doing next word prediction. So I'm not going to go too into the weeds about the LLM architecture, but what you need to know is just that the architecture is just the decoder only side of the transformer. And this enables these models to be trained on huge amounts of text corpora, such as the internet, published works, and whatnot. And obviously that's kind of led to this huge rise and boom of foundational LLMs that you can find off the shelf, commercially available online. But I also want to highlight kind of, this increasing scale over time. We have our GPT-1 model all the way up to what we're at 5 now, I think. But I also want to kind of highlight some models, foundational models that also have been created within the open source regimes, such as the Llama models, which will actually be the major focus of what we developed here in our thesis. And so, okay, talked about bio, talked about Gen AI. Why exactly do these two make sense? And I think there's two really strong reasons for this. The first one being that there's vast amount of knowledge already. I've already said this. There's a bunch of papers across decades, across so many different types of disciplines as well, because naturally biological materials are highly interdisciplinary. There's papers from biology, there's papers from chemistry, mechanics, engineering. And secondly, and kind of more importantly, I think, is something that's really particular about biological materials is that they're often described in natural language. So unlike other kind of scientific applications such as molecular design or protein design, there's no set symbolic representation for biological materials. And oftentimes also in mechanics, we're also describing things in natural language. And so for these two reasons, this is why we suspect that biological materials provide kind of this useful test bed for understanding language-based scientific Gen AI systems. So with this in mind, I now introduce the three core chapters of my thesis. I'll talk a little bit really briefly about what each one means. The first one is chapter two, bio-inspired LLM. This is a fine-tuned large language model for science, specifically specializing in biological and bio-inspired materials. The next chapter is then applying that model even further in a actual physical research workflow, understanding how we could use it to actually conduct real research and do materials design. The last chapter, similarly building off of bio-inspired LLM, but kind of shifting gears, we're moving away more from like text outputs from LLMs and into the multimodal regime. So understanding how we could use these large language models to actually create, or generative AI generally to create just more physical models. So we'll hop into chapter two now, bio-inspired LLM. The main kind of guiding question that we have here is, can domain adaptation enable LLMs to acquire scientific reasoning capabilities from legacy biological materials literature? And so the basis of this work was, we did this a long time ago. This was back in 2023 when kind of LLMs were really rising. And the main idea here is that we're harnessing the power and knowledge that's already embedded in a foundational LLM and fine-tuning it with domain knowledge. And so with that, we're introducing bio-inspired LLM, which is this conversational LLM fine-tuned on a literature corpus of mechanics of biological and bio-inspired materials. What this process looked like to actually develop, that first step is obviously obtaining that legacy literature. So doing text data mining, extracting over 1,000 full text research articles, collected using various means. From there, we do optical character recognition to extract all the text from those articles and to pre-clean any kind of artifacts. And then after that, we introduced the step that we call Q&A processing. So we found that after kind of doing step two, the data that we were extracting, that text was still very messy. It was still not well suited to be conversational for an LLM. So we did in step three was introduce Q&A processing, where we actually use an off-the-shelf LLM to help us further process and distillate the information into question and answer pairs, which also sets up the data well for doing supervised fine-tuning on an LLM. And then lastly, once we had that question and answer paired data, we then fine-tuned using a low rank adaptation strategy or a LoRa strategy. A LoRa strategy essentially freezes the entire original model and adds small trainable layers on top of it, such that you're only training those new small trainable layers. And because of that, you'll avoid any catastrophic forgetting. The model will retain its original knowledge while simultaneously learning new things. And so, okay, now that we have this model, how do you evaluate it? And this was a big question, especially in 2023 when a lot of this was not really quite figured out quite yet. But I guess kind of the knee-jerk reaction was like, okay, give it a test, make it a benchmark and see how well it does. And that's what we did. We made a new domain-specific benchmark for biomaterials, tested it, and as you imagine, it'd be like passing a question to bio-inspired LM, and it'll give us an answer. But we also employed this method called retrieval augmented generation, or also called RAG. And this pretty much changes things a bit. And the analogy I always give is that it changes it from a closed book exam to an open book exam. So the open book being this knowledge database where we're able to store the original corpus. When a question's passed to bio-inspired LLM, retrieved like relevant content will get passed as well in context, such that bio-inspired LLM will perform better downstream. And the way this looks on our actual benchmarks is that we find pretty much the best performance when we do this mixture of fine tuning as well as in context learning. And specifically, not only is RAG also improving performance, it's also allowing us to be able to retrieve and trace back sources, which, as we all know, is very important in terms of scientific research. I'm not going to be able to go through all of this, but here's a few other experiments that we did throughout this paper and this work, just kind of testing out the different ways that our model is able to accurately recall information about biological materials, from things like a materials classification task to, having the model even like test itself and generate its own question and answers. But kind of more interestingly than knowledge recall is hypothesis generation. So this idea that LLMs or Gen AI models could be used as this agent for creativity in scientific regimes. So these are kind of out of the box experiments that we're just like kind of really thinking about, just like, could a model help us do experimental planning? Could it help us generate data sets? Or even in this case here, could it predict mechanical behavior? If you knew me before MIT, I worked on jackfruit. This was a paper that we worked on pretty much in my last year at UCSD, but it wasn't yet published. So our model is technically not trained on this paper, and still I was really curious, could our model retroactively predict the mechanical behavior we saw? And it does so with kind of startlingly good accuracy. And then lastly, I'm going to show this kind of potential design workflow. I guess this can be known as like maybe a very early pseudo agentic system, where the idea here is not just using our LLM on its own, but also integrating it with other generative AI models. In our case, we're integrating it with Stable Diffusion, which is a text-to-image creator. The idea here is that a user could go to Bio-inspired LM, ask for some descriptive prompt. That prompt could then be passed to Stable Diffusion, which can then create these 2D images. Then these 2D images can be heat mapped and generated into a 3D model. And with that 3D structure, we're able to do some simulation experiments, and you can imagine it could form this like whole data-informed feedback loop, right? So, and then lastly, obviously, as time has gone on, these models have gotten better, a lot has evolved. But what was nice about kind of our methodology from the start was that it also co-evolved with the development of models. There's been minor changes where we pretty much expanded the data set and also introduced new fine-tuning techniques like reinforcement learning. But for the most part, this is the updated configuration that we use for our models going forward. Okay, so what did we learn? Here's the kind of key question again, where we're just asking what happens if you add domain adaptation to LLMs. We find that, yeah, when we do this, it enhances knowledge recall and hypothesis generation. And it's really interesting because these capabilities emerge simply from just exposing these models to scientific literature rather than doing task-specific training, which is what people normally do in scientific ML. And so, and then we also show that kind of potential for that integration into materials design workflows. So after this first project, we're again kind of just curious, okay, but how can we actually use bio-inspired LM into a real world materials research setting? And so with that, I now lead into our next project, our next chapter, which was actually this incredible collaboration that we did with some experimentalists based at NTU in Singapore. What you need to know about them is that they studied plant mechanics, but more specifically, these pollen-based materials. The idea here is that they're able to transform this highly abundant, pretty much biodegradable waste considered material into highly functional forms. So they're transforming defatted pollen to highly absorbent pollen sponges, humidity actuated pollen paper, as well as even printing on that pollen paper to be able to program how that pollen paper folds and actuates. Isn't that like super neat? So with this in mind, we had this Gen AI system that knew a lot about biological materials. And simultaneously, we had these excellent collaborators in Singapore that worked on kind of plant mechanics. And we were really curious about, okay, what if we built a Gen AI system that could really support this research? And so with that being said, here's what that system looks like. So I already showed this before, but bringing it back, RAGs here again. But instead of the knowledge database being kind of that original corpus, we're going to inject those plant mechanics paper to give our model that plant mechanics focus. And when we do stuff like this, we also do a bunch of sensitivity analysis. I'm not going to go into detail about that, but just know that you can tune a lot of these parameters to get the best performance. And you do that by like building out, again, domain-specific benchmarks, tuning those parameters, and yada, yada. But on top of that, there's a few other methods that we use. The first one I'll talk about is multi-agent interaction. So not only just having one LLM, but having the LLMs integrate and communicate with one another to do refinement, conversation. And another ability we introduce is tool calling. So, again, not using these LLMs alone, but instead integrating it with external tooling. So having it be able to call upon Semantic Scholar to do literature review. But still, one thing that we were really interested in was this idea of creativity in LLMs and being able to use LLMs as this creative agent. So we needed space for a new tool to be able to make this process more robust. So with this in mind, I'm going to side tangent a bit and talk about LLMs. LLMs are these probabilistic models where essentially what's happening is they're predicting the next most probable token. It's kind of hard to believe that the next most probable token or the next sequence of most probable tokens is also the most novel set of probable tokens. And so what people do when they're like, oh, let's make the LLM more creative is they'll increase this T parameter here, which is known as temperature. And what happens then is they'll flatten out that probability distribution, but still either way though, you're still sampling an LLM once, it'll give you some sort of response and it could be good, could be bad. We don't know for sure. And we're curious about it being a more robust way of doing this. So we thought, okay, let's do high temperature sampling, but instead of sampling once, let's sample many, many times. We'll sample so many times I'll have all these different candidates. And then from there, we can apply a selection criteria to help us filter down to the top most like notable candidates. And so we call this process hierarchical sampling, kind of as a bit of a homage to biological materials, but really it's nothing too crazy happening here. It's actually based on a lot of like foundational cognitive science theory about creative thinking, how it's this combination of when we often think about problems, we try to think divergently. We think about every single problem that could, or every single solution that could fix the problem, and then we'll converge on kind of what solution makes the most sense. And so, with this in mind, we now have this new tool for creativity, hierarchical sampling, and I'll show you how we apply it. So, a lot of this work was also kind of building out these algorithms and these systems where the users, all they have to do is just kind of input a prompt and they'll get whatever they're looking for. But what's really happening behind the scenes is this hierarchical sampling method. So, one of the ideas here was to be able to mine ideas. So, from a single query, we perform essentially a divergent generation phase where the model is sampled many, many times to get all of these unique ideas. And then simultaneously a convergent evaluation phase using LLMs, again, following some sort of criteria to narrow down and to highlight those top potential candidates. And with something like this, you might be wondering, okay, does this really actually help? And specifically for divergent generation, you might be wondering, is it exploring the design space better? And we find that it does particularly well, especially for kind of just the standard question of like, what applications could this plant material be used for? It's giving us ideas spanning from architecture to environmental monitoring to biomedical applications. And when you compare that to just a standard generation where we're just using an off-the-shelf LLM, you'll see that, yeah, it will hone in on sustainable materials because that's definitely the most popular kind of application and knee-jerk reaction. And we also do a bit more analysis on this too, where we also assess for novelty and really try to optimize this protocol that we built out. From there, here's kind of examples of a few prompts and top ideas that we get from our system. And what was really awesome about working with our experimental collaborators was that we are able to actually experimentally validate a lot of these kind of predictions. For instance, I talked a bit about how they can print on pollen paper to program actuation. One of the ideas that the model came up with was to print like a nature-inspired pattern or leaf venation. So our experimentalists went to the lab, got busy, and created that for us. And then simultaneously, another one here is this mechanical behavior coding prediction where we're asking the model for a coding that could that could freeze the dynamics of pollen paper. In this case, it predicted paraffin wax, in which our collaborators were able to, again, experimentally validate. Still, it's not very always seamless, and I think it's very important that we talk about that. So, in kind of these two experiments, we're giving the task of having this strip of pollen paper, and we're asking the model to predict how to print on it in order to actuate in a particular way. So, the first example is to induce like accordion-like folding. The prediction that the model made here was to print these hinges in alternating fashion on both sides of the paper. And I was like, that makes sense, feels right. However, when I went to the experimentalists and told them this, they were like, actually, we can't do that. It's just a fabrication constraint on our end. The pollen paper is just too glossy on one end, so it leads to very poor adhesion. And as a result, when it comes to actually actuating it, it doesn't quite work. And another example here is this one where we're trying to ask to fold this letter M. The model makes this prediction that is really, really based on kind of the theoretical values that are proposed in the original paper. It makes a lot of sense. However, in actual experimental practice, it didn't work. And kind of the experts came in and like quickly corrected it. And, I was pretty bummed about this result at first, but we kind of talked about it a bit more. And it was kind of interesting because the experimentalists gave us insight about how, kind of in practice, they knew from working on this material for many years that if they wanted it to fold in a particular way, specifically when it also has more than two hinges, the angles tended to need to be over exaggerated over the theoretical values. And also there's also like experimental variation from like the amount of pollen in the paper, how thick the paper is. But still, we wanted to highlight these two experiments because even though they're less so successful, they still reveal these kind of like structural limitations of our current system, which that literature trained models have this publication bias. If you've ever written a research paper before, I can bet that you probably did not document every single failure in that. And I think, or we think that that failure is a huge learning method for these models to get better, as they'll be able to understand that experimental intuition more. And so a few other experiments, I won't go into too much detail, but we do a few more kind of like mechanics-based ones. And then lastly, we introduce this other protocol. So, it's cool and all that our model can generate ideas, but being able to like go from an idea to a physical laboratory experiment is obviously non-trivial. So we designed this protocol where instead we can now robustly generate procedures using our system as well. And it goes through multiple phases where there's a phase of self-reflection, as well as a multi-agent refinement phase. And we do a good bit of analysis as well as kind of like a blinded human review and automated assessment to kind of show why this works the way it does. But I'm gonna move on for just time's sake to kind of show how we can actually combine these two protocols that we developed, have the model generate autonomously from the idea to also the procedure all in one go. And when we do this and we ask for, come up with a very novel design and simultaneously also that experimental procedure using our system and its protocols, one of the ideas is to design this water responsive adhesive using pollen. And what's really interesting about this is that, this pollen adhesive is not only a novel application of pollen, but this processing technique that is proposed in the procedure of using this tempo solution is also a novel procedure in terms of processing pollen grains as well. So because we had our experimentalists working on this as well, they're able to actually kind of integrate with our system, make any refining steps. But for the most part, they followed the procedure pretty much almost directly. And by doing so, they are able to generate this final microgel, which they reported having kind of a distinct grainy texture. And kind of because it was an adhesive, but they did this kind of like lap joint shear test where they just quickly checked to see, how much could it hold, and it held about 18 grams, which correlates to about 1.77 kilopascals of shear stress. And so, that's cool. But what happens if you take all that data that we just got and you feed it back into the system? And we're saying, okay, we did this procedure. Here are the results we got, and now we'll give it an objective function. In this case, let's improve that shear strength even further. And when we do this, the model and the system kind of provides a few different ideas. The main one being to increase the sodium bromide concentration. The researchers go back to the laboratory, do that. They also do a bunch more characterization for us because not only is this an adhesive, it's also supposed to be a water responsive adhesive. So they also did some like swelling ratio experiments. But when it came to shear strength, we saw about a fourfold increase in shear strength, to increasing it to about 7.86 kilopascals. And to put that in perspective, a fourfold increase over just one iteration is pretty prominent, but putting that in perspective to just other commercial products like a light duty arts and crafts glue stick, that those are on the scale of around 2 kilopascals or 10 kilopascals. So being able to develop kind of a material with this level of strength after only just two iterations was pretty exciting. I think it's pretty cool. Um, so with that being said, that concludes chapter 3. There's a few more experiments that obviously I don't have time to talk about today, but that paper is currently available in preprint. So again, kind of what are the main takeaways? Well, they're essentially having this more structured framework where we're integrating, our fine-tuned model, we're integrating RAG, we're integrating agents, hierarchical sampling, doing all this stuff to really enable more robust hypothesis generation and protocol generation. And because of that, and because we're working with our experimentalists, we're also able to demonstrate this end-to-end validation of AI generated idea to a physically experimentally realized material. And simultaneously, I talked about that example earlier about kind of where the model falls short, and that stuff that we reported in our paper. Because again, we don't want to hide the failures. We want to report that so that our models can get better and to learn exactly where the gaps are in the training data and simultaneously express this need for greater experimental context. All right. And so lastly, the final question is actually a bit more broad and very open-ended. It's just how can Gen AI be used to create 3D bio-inspired designs? So I showed this example earlier. It's from chapter 2. It's this like workflow where we're asking for these like text prompts and passing it through an image generator. And after kind of like doing that experiment back in 2023, we're always thinking about this step, particularly this one where we're just like heat mapping a 2D image into 3D, and we're like, there's got to be a better way of doing that. Especially also because the stable diffusion model is also off the shelf. It doesn't have any scientific grounding necessarily. It doesn't know anything about biological materials apart from kind of what it's learned from the internet. And again, bio-inspired LM also, it's this really cool knowledge oracle, creative person, agent. But really, if it's just outputting text and it says to make a lightweight material inspired by the cellular process of bird beaks, that really means nothing unless you're able to actually make that material or make that structure. So that's really what motivates this final chapter, which is about pretty much being able to translate textual description outputs into 3D. And we call this project Bio-Inspired 123D. Um, so with that in mind, with any kind of big AI project, of course, you're going to need more data. So for this project, we developed this new novel set of 3D parametric structures designed using Blender, which is this open source application. It's actually used for a lot of graphic design and animation, particularly, but it worked particularly well in our case because bio-inspired structures tended to be very complex, and we were also able to programmatically generate them. And we also align these new models with kind of these natural language cues, because like I said, biological materials are described in natural language. So for instance, we have our helical structures, our cellular structures, as well as our tubular structures. And so with this in mind, once you have a data set like this was such an open-ended problem, and we were constantly thinking, okay, what Gen AI models are out there? How should we represent this data? And also, what pathway do we take? Do we go from text to image to 3D? Do we go text to 3D? And so this project, yeah, we got started on it, and what really caught my eye at first was kind of these multi-view diffusion models, which are these image-based models that could generate kind of these canonical viewpoints of images. And so I was like, okay, let's try text to 2D to 3D. When I looked into that space, a lot of work involved a lot of point clouds. So um, just two experiments that we were looking into in investigations. We were testing out these two different models, one based in the diffusion model, the other being a vision transformer model. But kind of the main takeaway from this work is just that specifically this kind of like text to image to 3D pipeline ended up being just too dense and inefficient for what we're looking for. And specifically also passing it as a point cloud and doing that reconstruction phase ended up being a bit too unreliable. And so after that, I kind of walked away feeling a bit dejected, but I came across this model instead, which it's called Llama Mesh. It's a LLM that actually just goes from text to 3D. And the way it does it is you'll input this natural language prompt, the LLM will directly output the object file. And the object file is exactly what you see here. It's this like human readable output where it says V for vertice, followed by coordinates. And they'll do that for all the coordinates and followed by all the faces. And so we're like, okay, let's try this out. So we fine-tune this model using our own data set, specifically the helical models here. And we find that it actually works quite well. The model learns over time, it figures out how to build these systems. But the main issue, though, is that once we wanted to, move on to these more complex designs, that's when the object prediction became way too token heavy. Especially, this is listing all the vertices. So if you had a curved object, it has a lot more vertices. So it ended up being way too token heavy and just poorly suited for bio-inspired geometries. Still, though, it helped reveal kind of this potential for LLM driven 3D generation. So with that being said, that led to our final model and data selection, where the model we ended up using is based on, again, an LLM. And the data representation was not a point cloud, not like a dense representation, but instead actually code, code defined geometry, how we actually developed the models in the first place. So the models are developed in Blender, and essentially Blender uses a Python API where you're able to programmatically generate all these models using code. So instead of, generating the object file data, our user input for our LLM is just a natural language prompt. The output is the Python code, and then we're able to automatically validate that in Blender using a subprocess. The reason why we suspected that this would work a lot better is because code is a such more token efficient representation. The amount of tokens of a script is far less than a raw object mesh is. Simultaneously, a lot of LLMs have a lot of kind of existing code knowledge already. And lastly, because we were working within this kind of Blender backend, once you had that code and generated that model in Blender, you're able to export it into whatever kind of downstream mesh representation you wanted anyway. So like an SDL or a point cloud or whatnot. And so with that in mind, though, we were like, okay, let's see if we can fine tune LLMs to generate 3D bio-inspired geometries. But the problem was I only had 12 scripts. And if you ever thought about training an AI model, 12 samples is not quite enough. So from there, we're like, okay, let's build out this data set processing pipeline, kind of similar to what we did with Q&A processing, but let's think critically about base scripts and scripting and coding generally. The thing about coding is that there's never really one exact way that you can code something. You can change it up. And so that's what we did. We built out this distillation pipeline where we're able to generate kind of variants of the singular base scripts. And this would be variants that change the logic, use different functions, change the order. And from there, when we see that, when we do that, we find that, yeah, this definitely does help diversify the scripts. And we see that it semantically and syntactically helps spread it out, the scripts out across the embedding space. And then after that, we also do embedded reasoning. So not only kind of just showing these models, these canonical examples of scripts, we wanted to see if we could embed more reasoning traces so that the model can learn from them. For instance, it's kind of in this narrative step-by-step format where there is text that describes what each block of Python code does. And by doing this, our hopes were that the model would be able to learn rather than memorize canonical examples. And then lastly, because we're, generating all these new scripts, we're going to do a phase of quality check to actually verify that all the scripts run correctly using a Blender sub process. So again, here's kind of the overall methodology. The first step was developing that 3D data set using our domain knowledge. The next step was then to diversify and embed reasoning into all output scripts. And when we do this, we expand our data set from about 12 to 4.5K scripts. The next step then was to create natural language input prompts. And we do this in kind of like a Mad Lib style game where there's word banks and instruction templates to kind of generate how a user or human might kind of ask for a 3D structure. And then lastly, again, we're going to use this low rank adaptation strategy to do fine tuning. And specifically for this model, we ended up using Llama 3.1 3B instruct. For reference, 3B is probably as tiny as it gets for a large language model. So it's very, very compact model and trains in less than half a day. It's really quick. And so again, now that we had this model, which we call bio-inspired 3D, how do we evaluate it? Well, of course, we're going to integrate it with RAG. We're going to give it access to those base scripts so that it has a canonical example. And again, we're going to build out a new benchmark, but now focused on 3D bio-inspired structure creation. And if you're seeing patterns in my research, that's because you definitely are. And so for this benchmark, a lot of it was kind of really focused on these hard level questions where instead of, kind of just getting the model to reproduce what it was already taught in the data set, we wanted to see how far this model could actually, create new designs, extrapolate beyond the data set and beyond what it was taught. And so when we do this and we find kind of just generally with fine tuning and again, RAG, we get the best sort of performance for all these kind of unique and new canonical structures where they're combining motifs across domains. And then specifically though, we do see that that hard level is definitely where the model struggles for sure. So with that, we were really curious, okay, how do we improve bio-inspired 3D? And we're interested in maybe even try integrating it into an agentic system so that it goes through phases of autonomous iteration and evaluation before the user even sees the output. And with that, what happens in an agentic system is that you introduce these new agents. You have two agents just to help out fixing any runtime errors in code, a refinement agent to help edit any designs. But interestingly, we also introduce this evaluation agent, which is based on a vision language model or VLM. Vision language model essentially is an LLM, but it also takes in image tokens and inputs. And the main role of the evaluation agent is to essentially look at the rendered output, which because we're using Blender, it's really seamless. You just have, the JPEG of the photo of the object, and we'll have that evaluation agent come in and kind of give any feedback, rate and approve the final model. And we find that when we use this system, that's where we see kind of our peak performance, especially even considering it against state-of-the-art models such as GPT-4o Mini and GPT-5 Mini. Don't let the Mini confuse you because those models are still absolutely huge. And again, building kind of this amount of performance on just our 3 billion models is pretty spectacular. And we definitely see kind of this emergent behavior happening in hard level questions. The reason for that being that, with that autonomous refinement stage, these models are working together to collaborate and progressively make these models more correct towards the design prompts. I'm not going to go into all this, but a bunch more analysis, also all available in the paper recently published in AI for Science. But we're also evaluating a lot of the VLM selection, the effects of reasoning, performance compute trade-offs. But more interestingly is these pretty pictures of actually showing that we could 3D print these models and actually do downstream fabrication for these models. So all of these are novelly generated models by our system and then simply 3D printed on an FDM printer. But before I get too ahead of myself, I want to bring it back to where I started, which was just showing how we could integrate this bio-inspired 1-2-3 system with bio-inspired LLM. So we set up this workflow where essentially all the user has to do is input the name of a biological material. In this case, we have toucan beak, crab exoskeleton, and horsehoof wall. And from there, our kind of biological oracle kind of translates these using its knowledge about biological materials and their structures into these design prompts. With these text design prompts, we pass it through our system, which then generates these 3D structures, all novel in case. And then lastly, when we compare that to kind of the literature, we see a pretty strong agreement with kind of the schematics that have existed throughout literature and have been drawn up before. And so we see this kind of as a way and a tool of kind of building out this very compact and modular framework to really accelerate bio-inspired 3D design. So lastly, the main takeaways there are LLMs, again, kind of showing up again as this very flexible framework to be trained on code as geometry representations to do more efficient 3D design. Specifically, we show that the 123D agent system is really good at kind of enabling more reliable performance while still using significantly less compute than foundational models. And then also this modular pipeline where we're kind of introducing agents is also a way that we could introduce maybe more data set fidelity as well as real world constraints such as integrating like a manufacturing agent that could check the slicing of the 3D print or whatnot. And so with that in mind, that concludes kind of the core takeaways from each chapter. And you might be wondering, okay, that's cool, Rachel, but that's all about biological materials and what does this mean for science? And so that's kind of my next kind of main takeaway was like after working on all these projects, I was thinking about kind of the patterns we were seeing about how we could approach Gen AI for science. And I really think it kind of breaks down into these three sort of categories per se, or pillars even. So the first step being kind of the specialization or model training phase, where, this shows up in chapter 2 and 4, where we're building out these domain specific models, bio-inspired LM, bio-inspired 3D. We have this orchestration phase. So once we actually have these models, how do we best use them? This shows up a lot in chapter 3, where we're, again, applying bio-inspired LLM in the most robust way possible, doing in-context learning, doing structured prompting, hierarchical sampling. And then lastly, and I would argue probably most importantly out of all of these three different pillars is grounding and validation, which I would argue exists across all three chapters, where we're focused on, not only just generating AI insights, but really actually evaluating the reliability and connecting them back to physical reality. So whether that meant just building out domain-specific benchmarks as an expert to also doing experimental fabrication and validation, whether it be 3D printing a structure or physically fabricating a new pollen material, that is really where I see kind of this field moving towards. So with that being said, all of the work I've just showed today and model weights associated with this work is released open source and maintained in external repositories. I'd like to definitely recognize some financial support from National Science Foundation, as well as MIT Presidential Fellowship and the MIT Generative AI Initiative. This is my list of publications, references. This is just for fun. I had this PhD roadmap that I built in like my first year. And obviously it did not look like this in my first year. It looked very, very different, but just kind of outlining how these projects panned out, kind of getting that early project in on bio-inspired LM was huge to kind of build this foundational work and then hitting the ground running in 2024 with these two other projects leading up to my defense today, May 8th. So yes, thank you. That concludes the technical portion.
Maybe we can do 10 minutes questions. Does that sound okay?
Yeah. Should I do acknowledgements or should I wait?
Yeah, of course.
Yeah, okay. Yeah, I just absolutely, I need to acknowledge people. I'm sorry. And the first one I'm gonna acknowledge is Marcus. Yeah, so 4 years ago, I got into MIT and I sent a cold email directly to Marcus. I sent it before even visit weekend and like scheduled a meeting with him because I knew that like I wanted to talk to this man. I wanted to be in his lab. He welcomed me with open arms for sure. And one thing about MIT is that a lot of programs end up giving you kind of like a grace period to pick your advisor, like usually about like a semester or 6 months once you start the program. But I pretty much knew for a lot of certainty that I wanted to join Marcus's group when I first got admitted into MIT. And then simultaneously, I ended up committing knowing that I was going to join Marcus's group in the fall. So thank you, Marcus. I really appreciate your inspiration, your openness to exploring new research ideas. I think that was really foundational for a lot of this work, which is unconventional and exploratory. I really feel like I've grown in ways I never could have anticipated during my PhD. So thank you. Of course, I must recognize my wonderful committee, Ming and Rodrigo. Thank you both so much for your guidance and insight. They were super essential for this thesis. Like I remember even doing my TA about presenting this work for the first time, but you two have always been super receptive, super excited about it, and always ask such good questions. So thank you so much for your support through the years. Um, Absolutely, I need to shout out my collaborators. Again, Ming, super stressed, she's here in the back, as well as our collaborators, experimentalist at NTU. We have Shah, Jingyu, Professor Namju Cho, who led all these kind of pollen-based experiments. These efforts were absolutely instrumental in bringing that chapter to life, and it's really been a privilege to work alongside such visionary researchers and role models. Um, LAM, my lab. I guess I didn't define what it stands for, but it's my lab. No matter how big or small our overlap was, I'm really grateful for the thoughtful, engaging, and supportive conversations we shared. Special shout outs to Lee and Subhadeep, who are my lab office mates. Folks that I've shared many, many years with, like Jamie, Wei, Michael. There's many folks here on the right too who are a lot of alum who really helped me out when I got started here, including Jinze, Isha, and Andrew. And also early on, Nick and Sabrina were huge inspirations for me, where they paved a lot of this way about nature-inspired design for sure. And of course, I must shout out Marcin Moisesowski because he was a visiting professor in my first year, and him and I actually worked on kind of a lot of my early PhD projects that didn't end up in my thesis, but it was still very foundational and in getting kind of like my footing at MIT. And I really appreciated working with all these incredible people. Here's kind of a few examples of just how much fun we had over the years, from 2023 lab barbecue at my house to I think this is Jens's defense, this is MRS, this is when we carved a pumpkin with our logo, and these are more recent photos where I realized that we really like eating and going out to eat, I guess. um, and and so a big part about me too is remembering my roots and kind of the people that have helped me get to where I am today. And a lot of that is also recognizing Mark Myers, who is my undergraduate advisor, as well as who was at the time Ben Lazarus, who was the PhD student working with Mark Myers, and both of them like took me under their wing and absolutely introduced me to the field of biological materials to begin with. I worked with horses, I worked with jackfruit, I worked with like aeroprima fish scales, and I really believe that, yeah, they really helped instill kind of those research foundations that made a lot of kind of my achievements in the PhD really possible. Of course, I want to thank my family. A lot of them couldn't be here today. Many of them are online, I think. But I really want to shout out my mom. My mom's here, right next to me, right here. She raised me pretty much as a single parent after immigrating here without even a high school diploma. And when I told her I got into MIT, she had never even heard of it. Um, but through all of her sacrifices, she really gave me opportunities that I will never take for granted. I'm super, super happy to be here right now in this moment to make her proud. Um, to my sister Amy and her husband Jed, thank you for instilling in me such a strong work ethic and for always surrounding me with so much laughter and warmth. To Kai, who was born recently, he made me an aunt. He is given me such a huge perspective on the next generation of learners. And yeah. To my brother Megan, or sorry, my brother Brian and sister-in-law Megan, thank you so much for all the time we shared. I absolutely loved all of our Boston and New York trips, and I just thought that this journey was made so much more fun with you two around. So thank you. This is also my childhood dog, who is very old now, but still love seeing him when I visit. And this is my grandmother, who's in her 90s now, but still remembers, so sharp, still remembers pretty much the dates of when I visit, and I absolutely see her every single time. And I'm super lucky to have also second families too. Thank you specifically to the Karens and the Dealvas, many of which who are in this room today, for the constant support. You all are always so excited to hear about my research. And I feel so lucky to have had you in my corner cheering me on, especially today. Thank you so much, everyone, for being here. Um, to my East Coast friends, yeah, I don't even know where to start. You all made this place feel like home. So much so that it's really hard to leave, and partially why I decided to stay in Boston and get a job here. So yes, everyone from the start, Pete, Tatum, Jessica, Bella, Roland, Maggie, Avi, Liam, Alice, Satya, and Stravia, thank you all so much for just making the East Coast feel like home. And then, of course, also shout out to our neighbors, Eric, Kirsty, and Owen, for being wonderful neighbors. Um, and and of course, I again do not forget my roots. My West Coast friends are absolutely incredible, who also flew 2,000 miles to be here today. Thank you so much for being here. You all made every goodbye so difficult. But I never forget how much you believed in me and brought so much joy in my life, even from afar. So thank you, Jane, Rianne, Natalie, and Adam. Oops. And then lastly, also just the broader DMSC community and the folks that I've been able to meet through MIT from just like, this is our third year cohort photo, this is GMC, the folks from the Forge, our committees, like all of these moments, Amir Sharash as well, like all of these moments are just so incredible. I met so many incredible people being here at MIT that I will absolutely never forget and would love to stay in touch and grab coffee all the time with. Um, and kind of shifting gears a little bit, I've been incredibly, incredibly lucky to have had the chance to travel internationally during my PhD for conferences and talks. Growing up, I didn't really have opportunities like this, so many of these experiences were my first, but they've been truly memorable and meaningful, and they really have helped me expand my perspective on the world and on my work in ways that I'll absolutely carry with me going forward. Thank you, Subra, as well, for two of the three international invitations. I felt like I left every trip as a changed person. Um, God, okay. And last but not least, my fiance, Jesse. We've been together before even our first day at undergrad at UCSD. We graduated UCSD together. Sorry, I looked at him and now I'm like, we came to MIT to grad school together and we even got engaged last year. So if you do the math, that's about 8 years. And you've been my constant through it all, throughout my education. Thank you for believing in me, even in the moments when I struggled to believe in myself. Your support, patience, and steadiness carried me through this entire journey. And people say like, "Oh my God, you've done so much, Rachel. You've accomplished a lot. Whatever. Maybe I have. I don't know. I probably have accomplished a lot in my life and career, whatever. But truly, my like favorite and most proudest accomplishment is finding you and building this life we have together. So, thank you, and thank you everyone for being here today. I so appreciate it.